Welcome to this live stream. I'm delighted to be back again with Professor James Shapiro. How are you doing, James?
I'm doing very well. And you?
Good. Thank you for joining me again on the live stream. So today we're going to be going over a paper that you have published alongside Dennis Noble. And I'm just going to be sharing my screen. So if anybody wants to read the paper is called What Prevents Mainstream Evolutionists Teaching the Whole Truth About How Genomes Evolve. So to begin off with, I guess the easiest question is to do with the neo-Darwinian modern synthesis. So you argue that molecular biology has undermined the neo-Darwinian modern synthesis rather than supporting it. Can you elaborate on the key molecular discoveries that you believe have discredited the core assumptions of the modern synthesis?
Okay, well, will be a long answer because so much has been discovered since the time of the modern synthesis in our understanding of how genomes are organized, how they function, and how they change. The first point is the whole idea of genomes are not made up of genes as the basic units of life, to quote Nobel laureate George Peele. Instead of units, all genetic loci are systems composed of different elements that include complex arrangements of coding and non-coding sequences that allow the cell to control expression of the encoded information by transcriptional regulatory molecules recognizing specific sequence motifs. The oldest and maybe simplest example of a regulated genetic locus comprising multiple regulatory sequences and coding elements is the E. coli lac operon, which has 3 operator sequences for binding repressor tetramers, 2 promoters for the lac repressor and for the function lactose utilization enzymes, and 1 CRP binding site to make sure that operon expression only occurs in the absence of glucose. I gave the technical terms, but the point is that the sites are there to control the expression of the proteins involved in lactose utilization. Monod showed in 1941 that E. coli can discriminate between glucose and lactose. Often, transcriptional control involves interactions between different regions of the genome, as occurs in the topologically associated domains, or TADs, that bring enhancer elements close to transcription start sites in eukaryotes, which are often on a different chromosome. So none of these elements were present in the neo-Darwinian modern synthesis. For them, it was a simple matter that 1 gene encoded 1 character. You have to give me a second to
Sure.
get back to my notes.
No worries.
So another basic tenet of the modern synthesis is that evolutionary variation or change occurs by accidents, by the accumulation of random accidents. If were that to happen, it just doesn't make sense because there's a vanishingly low probability of generating any significant genomic information that way. The likelihood of making a functional genetic element is the multiple of the probabilities at each nucleotide position in that element. This number becomes impossibly small at 2 or 3 nucleotides at an optimistic mutation rate of 10 to the minus 5 per nucleotide per replication cycle. 2 nucleotides would be 10 to the minus 10, and 3 nucleotides would be 10 to the minus 15. Moreover, we now know how the proofreading activities of replicative DNA polymerases and post-replication repair of any base pairing mismatches that make it into fully replicated DNA, reduce the level of mutations resulting from replication errors by about 6 orders of magnitude. All the supposedly spontaneous mutations are far more likely to be the results of cellular biochemical activity. Mutated DNA polymerases, which don't proofread because they're used to replicate across chemical damage in the genome. Base modification activities like cytidine deaminase, which is used for somatic hypermutation in antibody maturation, or one of several mutagenic repair systems for coping with replication blockage, DNA damage, and double-stranded breaks in the genome. We are just beginning to learn how powerful some double-strand break repair functions can be in generating genome novelty, including the insertion of untemplated, completely de novo sequences in the repair junctions. In other words, the overwhelming majority of genome change results from biochemical action. Another point of divergence from the modern synthesis is that important evolutionary changes do not always occur by variation in a single organism's genome, as the modern synthesis supposes. As researchers apply DNA sequencing to trace the lineage relationships between similar species, they sometimes discover DNA encoding important adaptive functions without any source in the genome of ancestors. Instead, the adaptive DNA could often be traced to completely unrelated organisms, and it must have been acquired by a process of horizontal transfer. That's transfer between different organisms. For example, beetle taxonomy is well established, and some clades include lineages of plant-eating herbivorous species. Living off such a vegetarian diet requires enzymes to break down complex plant polysaccharides, which are not found in most beetles. The herbivorous beetle lineages acquire the DNA encoding the necessary enzymes from different identifiable species of bacteria or fungi. There are also cases like the white flies that hijack a plant detoxification enzyme to protect themselves from plant toxins. Such horizontal DNA transfers occur in all directions between different life forms and are appearing to be more frequent as time goes on. So that's another element of well-established element of evolutionary change, which is not included in the modern synthesis.
Okay.
Another point of difference is that many organisms have protein coding loci that are not continuous. They are made up of exons, expressed elements, separated by introns, inserted elements. It was a huge surprise when some of the earliest DNA sequences revealed the unexpected phenomenon just under 50 years ago. To encode the protein product, the RNA templated by the introns has to be removed or spliced from the initial transcript produced to produce an mRNA that can be transmitted to the ribosomes. This continuous coding helps us understand how proteins frequently evolve by rearranging blocks of polypeptides known as domains, which have their own properties, such as binding to a particular DNA sequence, binding to another protein, or executing a particular biochemical process. In this way, proteins can undergo a Lego-like process of rapid evolution by exon accumulation rather than the slower accumulation of single amino acid changes specified by the modern synthesis. This process of accumulating and recombining domains was well illustrated in the 2001 Nature paper on the first draft human genome sequence. Often we now know the intronic sequences, the ones that were spliced out of the RNA, can often have their own effects in the cell, target largely as active regulatory RNAs after splicing. The realization that genomes don't just encode proteins, but also a huge range of functional so-called non-coding RNAs, or ncRNAs, many of which contain transcripts of repetitive DNA, elements, tells us that there are still a lot we have to learn about how cells control their genomes, and also the notions of junk or selfish DNA, which had no function but to perpetuate themselves, are simply incorrect. In the past 80 years, we have learned and are still learning about a series of different biological functions that create significant genetic changes and have proved to be of evolutionary importance, but they are not mentioned by the modern synthesis, which continues to insist on the accidental nature of genome change variation. These biological agents of genome change include symbiotic cell fusions for acquiring and integrating DNA from other cells or from the environment, elements that mobilize DNA between cells, sequence-specific recombination functions that generate useful genetic constructs like multiple antibiotic resistance array in bacteria, viruses that endogenize by inserting into cellular genomes, transposable and retrotransposable elements, reverse transcription, and systems that generate coordinated structural chromosome rearrangement simultaneously at multiple genome sites. This is called chromoanagenesis, which means chromosome rebirth. So all of these things are new and have no place in the modern synthesis. Chromoanagenesis in particular is only since 2011 that we've realized that this is going on, and that's as a consequence of sequencing whole genomes and finding out that they have these coordinated chromosome rearrangements of different kinds. So those are all the reasons that I think the neo-Darwinist modern synthesis is just out of step with what's been going on in molecular biology and genomics.
Thank you for that comprehensive answer, James. In regards to chromoanagenesis, for anybody who's interested, I did a separate live stream with Professor James a few months ago, and I'll leave the link to that in the description for anyone that's interested.
Good.
You mentioned that the idea of DNA being an intrinsically faithful self-replicator is a myth. What implications does this have for our understanding of heredity and evolution?
Well, DNA cannot replicate itself. It takes a living cell to replicate a genome, to produce the chemical precursors, to polymerize the strands, to repair, proofread, and physically organize the replicating genome. That means that genomes are subject to all the biological functions mentioned at the end of the first question. Heredity and evolutionary change are fully biological functions. Biology is not random. It changes itself when confronted with new conditions. We know that evolution can occur quickly in response to certain stimuli, such as carcinogenesis, chromosome breakage, and interspecific hybridization. So that's all I have to say about the DNA being a replicator.
Sure. Just before we move on to the next question I sent you, I just wanted to prod you about the idea of genes being replicators and the body being vehicles. This idea is directly contradicted by the fact that DNA is not a self-replicator.
Yeah, that was based upon a theoretical speculation in the book What is Life? And I'm having a senior moment about the name of the author. He was a quantum physicist. Schrodinger's book, yes. And Richard Dawkins picked up on that and developed a whole philosophy of evolution and of more than that, of natural behavior and so forth out of that idea. But DNA doesn't replicate itself. And it's part of the cell. It's a kind of database for the cell, which allows it to make a range of proteins and RNA molecules that it uses during various forms of life. I just think that that's a cute notion, but it's just not applicable to the reality of what goes on in cells and multicellular organisms.
Yeah. Thank you for that answer. And in your paper, as you highlighted in your first answer as well, you mentioned symbiogenesis and horizontal DNA transfers. How do these processes change our understanding of evolutionary mechanisms when compared to the standard neo-Darwinian modern synthesis, in which it's specified that gradual accumulation of random changes is what causes evolutionary change? So we're talking about timescales.
Yeah, well, there's two general significances to symbiogenesis. Other forms of symbiotic associations and horizontal DNA transfers. The first is that evolutionary transformations can occur rapidly. Symbiogenesis is an event which takes place in the lifetime of two cells. Horizontal DNA transfer may take place over a longer period of time, depending on how it happens, but it's still a rapid change for the cell which picks up the acquired DNA. It acquires an already evolved function, which it needs to live in a new ecology. So evolutionary changes can occur quickly. And the second is that the nature of the changes for symbiogenesis, symbiosis and horizontal transfers, both depend on the local biological environment. They're not just internal to the organism, they depend upon other organisms being around them. So symbiogenesis will have different results depending on the particular partner, the organism which is acquired by the host cell. For example, the symbiogenetic event at the base of eukaryotic evolution involved an aerobic proteobacterium, which conferred the ability to use oxygen for energy metabolism on the anaerobic archaea, another prokaryote, that engulfed it. Well, the symbiogenic events that produce photosynthetic organisms, photosynthetic eukaryotes, I have trouble here with my notes. Yeah, well, the symbiogenetic events that produce photosynthetic eukaryotes involve eukaryotes involved in engulfing other prokaryotic cyanobacteria, which are photosynthetic and are the basis of all the photosynthesis that we see in eukaryotes, or photosynthetic eukaryotic algae, which themselves had previously engulfed cyanobacteria. So that's a sort of two-stage symbiogenesis. And symbiogenesis and symbiosis are continuing to this day. So organisms can quickly change their properties by picking up another organism and internalizing it into the cell. Likewise, the acquisition of horizontally transferred functions depends on the genomes of the organisms present in the immediate environment of the evolving organism. We know a great deal about the mechanisms and outcomes of horizontal transfers in bacteria, transfers which have been tremendously important in the evolution of multiply antibiotic-resistant superbugs that threaten our ability to cure many kinds of infection. We know much less about horizontal transfer in eukaryotes, although there have been experiments showing that yeast, algae, and plants can take up naked DNA from the environment, as many prokaryotes are known to do. DNA transfers by viruses or virus-like particles between prokaryotes are routine, but have also been reported for exchanges in eukaryotes between insect species, mammalian species, and from bacteria to mammals. There is also a growing literature on direct cell-to-cell contact, facilitating DNA transfers from bacteria to all classes of eukaryotes, ranging from yeast and other fungi to plants and animals. As far as I know, we have no empirical information about how horizontally transferred DNA is integrated into the recipient genome, but it has to include biochemical functions that join DNA molecules together. And none of that is included in the modern synthesis.
Thank you for that answer. And just to emphasize what you've said about horizontal gene transfer in terms of bacteria, we've known this for at least 50 years now in terms of the large-scale application of antibiotics and the rapid evolutionary changes that we've seen in bacteria where they share the resistance DNA.
Yes, absolutely. I'll come back to that later.
Sure. So the next question is, you discussed the role of stress responses in activating intrinsic genome changes. Can you explain how this concept relates to the clinical outcomes in oncology?
Yeah, this is something that I became very interested in, and we organized a cancer and evolution symposium about 4 years ago, and it turned out to be extremely informative. Cancer is actually, it's the case of somatic evolution. That means evolution without going through the germline. And it takes place in the body. And tumors and cancer cells in them evolve over time. Generally, they arise from events which vary from cancer to cancer. But once the tumor is formed, it can still evolve, and it generally goes from less malignant and localized to more malignant and metastatic. One of the factors that can affect this evolutionary process is chemotherapy. A number of lethal chemotherapeutic drugs were designed to kill the maximum number of tumor cells. But treatment with these drugs almost invariably resulted in formation of a lethal therapy-resistant malignancy within the next 2 years after therapy. So it gave some short-term relief to the patient, but long-term, it didn't work. Looking at the cancer from an evolutionary perspective, it is evident that these lethal drugs triggered the tumor cells to evolve rapidly to greater malignancy and metastatic potential, as well as to resistance to anti-tumor therapies. In other words, the attempt to eradicate all the tumor cells converted what was often a bearable cancer burden into a fatal disease. Oncologists are learning that their strongest anti-cancer medicines may be worse for their patients than more gentle treatment of tumors as chronic diseases, because tumor cells are alive and can execute an evolutionary response to chemical assault. So that gives us a different way of thinking about how we deal with cancer.
Yep.
We realize it's living and can change. we approach it differently.
Yeah, absolutely. And you've been working alongside Noble and I believe Perry Marshall in regards to this revolutionary paradigm shift in terms of cancer. And this is something I've covered with Perry previously on the channel as well. So thank you for that answer. You note that the active view of evolution emerging from genomics is closer to the ideas of Darwin and Lamars. How do you think their theories can inform current evolutionary biology in light of modern discoveries?
Well, this is interesting. Both Darwin and Lamar believe that life experiences influence the inherited properties in an organism we pass on to its offspring. That's commonly known as the inheritance of acquired characteristics. Darwin even proposed the existence of particles, which he called gemmules, which could be the carriers of such lifetime experience. We now know about the existence of microscopically visible particles known as extracellular vesicles that carry cargoes of protein, RNA, and possibly also the DNA to germline cells, especially to sperm cells. And these transferred RNAs are needed for successful development of the fertilized egg into a viable embryo. So there is no Wassermann barrier separating the germline and soma in animals. And there are a number of experiments in nematode worms and in mice demonstrating the transgenerational inheritance of acquired characteristics. For example, a worm can be trained to avoid certain bacteria when it's growing near it. And without being trained, its progeny have the same avoidance response. So that acquired characteristic can be passed on to the next generation. Naturally, all germlines develop from somatic tissues in organisms other than animals, like fungi, microbes, and plants. So there is not an issue of separating the germline from the soma. The question is to learn how and to what extent experience affects the nature of evolutionary variation. And I'll have some more to say about that in a little bit.
Thank you for that. The next question is, your conclusion indicates that outdated evolutionary concepts in medicine can lead to ineffective treatment strategies. Could you provide examples of how a better understanding of evolution could improve medical practices, particularly in regards to cancer?
Yeah. In oncology, the realization that chemotherapeutic treatments can accelerate tumor evolution to a more deadly form has led some practitioners to avoid trying to wipe out all the tumor and apply what they call adaptive therapy, which aims to slow the growth of the tumor without triggering its evolution into a lethal malignancy. So that's a big change in treatment philosophy in cancer. In infectious diseases, the goal should be to avoid the overuse of antibiotics so that transmissible resistance does not render them useless in the future. And this is a major problem around the globe now. We grossly underestimated the capacity of bacteria to evolve widespread resistance when we first started using antibiotics. The theory was that, according to modern synthesis, antibiotic resistance was the result of isolated mutations. There's no idea of transmissible resistance or multiple resistance. The result is that many life-threatening infections are no longer curable with antibiotics. And we have to find other treatments, such as using antibacterial viruses that are called bacteriophages or phages to kill bacterial pathogens. And phage therapy was a new idea at the beginning of the 20th century, but it's come back into fashion because we need alternatives to antibiotics to treat bacterial pathogens. I think it's interesting that a failure to understand the evolutionary principle led to such an outcome.
Yeah. And this also explains why after cancer treatment, when it's assumed that the cancer has gone away, that it comes back much more aggressively the second time around because of this rapid evolutionary process. And this likewise why antibiotics, the overuse of them, is actually ineffective, which is why we need a new penicillin.
Yeah, well, maybe the phages will give us a way of dealing with specific pathogens. If we can find enough viruses or phages which kill them, and we can develop those kinds of therapies. There may be other ways to do it. People are looking for different kinds of antibiotics, but I suspect that any antibiotic which is developed will ultimately the bacteria will ultimately come up with resistance mechanisms, and those are subject to transfer.
Yeah. Yeah. Well, at least from your perspective, we understand the arms race a lot better than the traditional way, in which we assume that there's just point mutations, which leads to the accumulation of new functions.
Right. Yeah. And hopefully we'll be, the medical community has become wiser in its prescribing behavior.
Absolutely. You mention a campaign to maintain the neo-Darwinian modern synthesis status quo within mainstream evolutionary community, within the mainstream evolutionary community. What do you think are the main barriers to accepting these new ideas in biology, and how can they be addressed?
This is an important question. I think the mainstream evolution community has been unscientifically insistent that Darwinism had all the answers about evolution. It's always a hazard in science to think that you know the truth, the absolute truth, and you don't have to look any farther. They have failed to teach students the full picture of what we know now about the lessons from genome sequencing and from experimental studies of genetic variation. The last time a colleague and I looked at standard evolutionary textbooks 3 years ago, that was Dennis Noble, for example, there was no mention of Barbara McClintock or the important role the transposable elements, or TEs, have played in evolutionary transformation. This is a major error. For example, TEs provided key coding and transcriptional networks that made the vertebrate adaptive immune system a powerful generator of antibody diversity. They also rendered viviparous reproduction possible in mammals, and they provided 63% of the primate-specific regulatory regions. So we wouldn't have evolved without transposable elements, yet the mainstream evolutionists ignore them. The same willful ignorance applies to all of the other cellular systems living organisms have used when genome change was necessary for survival. The way to deal with this refusal to accept the results of molecular genetics and genomics is to provide the public, especially students in secondary school and postgraduates, with accurate information on what has been learned since the middle of the last century. And we're looking into doing that so that people can have access to up-to-date information about what happens in evolution and the many things that would be surprising to them, but that are very well documented. This will, the problem, one of the problems is that academics and teachers are reluctant to go against the modern synthesis. They'll get condemned for doing that. And they don't have access to all of the research information and are not yet able to put it together in the same way.
Right. A follow-up question I wanted to ask you in regards to this. Do you think the resistance to changing the status quo of neo-Darwinism, the modern synthesis prevailing, is that the people who put it together were of such high caliber and such giants of biology, such as Crick and Huxley and others? Do you think it's the status of these individuals which has led to the status quo?
Well, that's a big part of it, because they've all been hailed as heroes and big shots. But discovering one thing doesn't mean you understand the whole, everything about that subject. And you can make errors, because Crick's idea, for example, of the dogma of molecular biology. It's an interesting word for science. Dogma was that information comes from DNA to RNA and from RNA to proteins, and information cannot go in the other direction. But actually what we've learned is proteins can do a lot of change in DNA, and they do it all the time. So the modern synthesis only has a one-way flow of information. And that's true, that information flows that way, but it doesn't say that information doesn't flow in other directions as well. So I think the prestige of these people has played a big part in keeping contrary information and results from the general public.
Yes. And another follow-up question in regards to this, your mentor, Barbara McClintock, who won the Nobel Prize for her discovery in terms of genome reorganization, what we found in the study in 2001, the Nature paper, was that a large part of the genome was actually evolved through this type of process. So we've known this for a very long time. So it's not that there is a lack of scientific evidence, it's that there's a lack of willingness to change.
Well, yeah, the problem with that for the modern synthesis way of thinking was they thought that each gene had to be unique. They could be related, but they were different. And here we had genomes like ours, which have millions of copies of repeated sequences, transposable elements. And Dawkins' first book, significant book on the selfish gene, said, well, they can't be genes because they're all alike. We now know that they play an important role, not only in making genetic change, but in encoding functional RNA molecules. And we don't quite fully understand how that works yet, but because this is pretty new, but it's a reality. And as I said, the term selfish or junk DNA, this repetitive DNA, doesn't make any sense. And once we realize that this DNA participates in encoding useful molecules in the cell, we can overcome that mistake and begin to study them in more detail.
Yeah, absolutely. The next question, how do you believe the continued propagation of the modern synthesis affects the general public's understanding of evolution and biotechnology, particularly with tools like CRISPR?
Well, CRISPR and similar targeted genetic engineering tools are useful tools for what is called genome editing. But we have to note that that makes what are called microevolutionary changes that do not create new species, but change individual characteristics. CRISPR will be used, is proposed to be used like repairing hemoglobin encoding DNA in someone with sickle cell anemia, or inactivating endogenous viruses in the pig genome, so pig organs can be transplanted into human recipients. There are attempts to increase the power of genome editing by doing multiple edits at the same time, called multiplexing the edits. However, to create genuine novelty species and make a macroevolutionary change, which is distinct, we need to do what our ancestors did in the past to create new crop plants, which is to cross two similar species and thereby activate the speciation functions in the hybrid progeny. These include activating the movement of transposable elements through the genome and the formation of chromosome rearrangements. And these changes will both alter the adaptive characteristics of the new species and prevent it from mating with its parent species. This process is called hybrid speciation and has given us wheat, rye, sorghum, cotton, potatoes, canola, and many other economically important crops. You call canola, I think you call rapeseed in Britain. I suspect that hybrid speciation may prove more fruitful than genome editing for many purposes, such as developing organisms that fare better under climate change conditions. So there's a lot we can learn from doing interspecific hybridizations and seeing what emerges from that. And we haven't, hasn't been studied systematically. Just what we know the changes occur, but we don't know if there's anything about them that is biologically relevant in the way they do that.
Yep. The next question, what do you envisage as the next steps for evolutionary biology in light of the genomic data and molecular revelations? How can the scientific community better integrate these findings into educational frameworks?
Well, the next steps for evolutionary biology, I think, is to study what conditions, besides interspecific hybridization in cancer, will trigger macroevolutionary changes and start the speciation process. Active research into chromoanagenesis, that is the multiple changes in the genome structure that occur in a single cell division, giving genome-wide structural changes, offers some promise for doing that. Another focus is to begin analyzing how sensitive the results of speciation changes are to organismal experience. Remember, I discussed how we now know that life experience can influence heredity, as both Darwin and Lamars believed. For example, some groups of butterflies are known to evolve by hybrid speciation, and it should be possible to carry that out experimentally under different illumination conditions to determine whether newly evolved wing patterns are affected by ambient light and colors. So we can do experiments that would not be considered under the modern synthesis. They wouldn't have thought that anything that happened to the organism could influence the way it changed. But we're liberated from that limitation now. So we can begin to ask those questions. And they may come to nothing. On the other hand, we may start to learn something more about this process of genome change and speciation.
Thank you for that answer. The last question. What advice would you give young scientists and researchers who are interested in studying evolution and its mechanisms in a way that aligns with the latest genomic discoveries?
Well, I would advise them to learn molecular biology, taxonomy, and genomics as the first step, so that they know what has been discovered. And then I would say pick an organism that fascinates you and carry out research on what conditions make it change without worrying whether your conclusions fit with anybody's fixed ideas on how change should occur. Never assume that our knowledge is complete. There are always surprises, unexpected discoveries, and you have many tools available for studying the molecular and cellular processes underpinning some of these hereditary modifications. I think the history of genetics and evolutionary biology is an object lesson in the fact that one never has the whole truth under control. You only know what you've learned so far
Yes.
with the technology and methods available to you. Nobody can predict what an unexpected or surprising result is going to be. And I think if you had told, well, maybe you still tell modern synthesis people about many of these discoveries about symbiogenesis or about horizontal transfer or about transposable elements, they will still dismiss them. Certainly they would have dismissed them in the 1950s as fanciful, and yet they've proven to be true. And they've given us a completely different view of how life changes.
Absolutely. Absolutely. And I guess they should adopt Darwin's spirit, because, he evolved his thinking in his books, the later editions of The Origin of Species. He, was a lot more open to Lamarckian ideas. So I think that's the right spirit to have, not to start off with preconceived notions and try to get the evidence to align to it.
Yeah, I think he was open, I think, to inheritance of acquired characteristics from the beginning. I think it's in the first edition of Origin of Species. But in the sixth edition, he recognized that there were mechanisms of change that happened to organisms which were completely independent of natural selection. And that was a large concession by him, because that was what he thought was his key idea, was natural selection. So he was very rigorous and honest in that way. And I think that spirit is one that should be adopted.
Yeah, absolutely. And I guess it's also important to clarify that when we're speaking about neo-Darwinism, it's not something that we can necessarily say Darwin would have endorsed, because his ideas were way more nuanced and more Lamarckian, if you like.
Yeah, I think what he wouldn't endorse about the neo-Darwinism is the dogmatism that it developed, and the exclusion of other arguments. The treatment, for example, of people like Richard Goldschmidt and then Margulis, saying that there are other ways for organisms to evolve by random mutations in natural selection, and that they had the basic truth, and that they were the only ones who had it. And I think that's what has been a big problem in terms of educating young life scientists.
Absolutely, absolutely. Thank you so much, Professor James Shapiro, for your time today. A couple of months ago, when I asked you what you were working on, you said you were working on a book to simplify your ideas for the general audience. Is that still in progress?
Well, I think this exercise was part of it. What are the things that have to be explained? And I'm hoping to get down to writing it in the coming year. It's a challenge to take so many technical and subjects and explain them understandably to the general public. I think my answers for this podcast are not necessarily clear to people who don't know about biology and molecular biology. But there are ways to explain these things, and I have to find them.
Yeah. And Richard Dawkins, despite him being on the other side, his success is based on simplifying very complicated ideas and making them quite accessible and lucid to the public.
Yes, he's a fine writer, but the arbitrariness of his ideas, is not to be admired. I think sometimes it's simpler to oversimplify things and put out a story which is a consistent story, but it doesn't necessarily coincide with the empirical data.
Absolutely. Absolutely. So on that note, thank you so much, Professor James Shapiro, for your time, and I look forward to future live streams with you.
Good. Thank you.